All right, here is a quick overview of the topic of measuring biodiversity. So when we measure biodiversity, there's three ways we can do it, depending on what our focus is. One of them would be we could focus on how diverse are the different types of habitat available for organisms to live in. So if an area was just a big field with no trees, it's not going to provide that many different habitats, and that's the case. It's not going to offer many opportunities for different organisms to exist there. So just by the fact there's not many options for places for organisms to live, it's going to make it very unwelcoming for a lot of organisms. So, it doesn't necessarily to be huge differences in habitats. It could be a field, and that field might contain trees. It might have a little pond there somewhere. It might have hedges. These small little different small things will make a big difference. A hedge is going to provide really good opportunities for insects, birds to all live there. If those hedges get taken away, there's nowhere for them to live, and that area suddenly is going to lose a lot of species. So habitat is one way we can do that. Then we get even more information. Let's actually look at what species we have. And we can do one thing. We can count the different number of species, and we call this species richness. And that will give us some information, but it's not enough to tell us if the area is biodiverse or not. It's important that we count the populations of each species as well. Is the population sizes of all the different species similar? If they're similar, that tells us that the environment is very biodiverse. If, however, there's only one or two species which are the dominant number, that's a sign of low biodiversity. So some of the behaviours that we're doing as humans with farming, with monocultures, we're really driving down. There's very few species available in that area, and there's certainly not much species evenness if there's only one or two types of plant in that area. So when we take into consideration species richness and species evenness, we are using this equation, and this equation is our Simpson's index of biodiversity. So we're going through this. So there's the sum of the number of one species. What's the total number that was counted, divided by the total number of all the species you found in that area. Okay, so then how do we actually go about measuring or getting some quantitative measures of the different organisms that we find? Well, there's two main ways we can sample. One of them is random, which its aim is to eliminate any bias. You are just going into a field, you are randomly selecting where your quadrat is put down. You don't get to choose where it goes. You might want to set up some coordinates, and the coordinates tell you where to put that quadrat. And then whatever's in that quadrat, you count it either individual numbers or maybe a percentage cover of that quadrat. The other way is non-random, which is, there's a bit more bias there. Opportunistic, you may be interested in a particular species. So, for example, you might see, like, I'm really interested in what's going on with this interesting looking bird. I want to be going out of my way to find out about this one. So you've got an agenda really to what you're looking for. You're actually going out of your way to find a very specific species. Then we've got stratified, and this is being used because you know something about that habitat. You are aware. If we go back to this habitat diversity, you might be aware that the field that you're going into contains forest and it contains a bit of a meadow, and you might be concerned that if you were to just do random sampling, just out of bad luck, all of your random coordinates may only put you in the meadow area, and it won't represent what was happening in the trees. So you are making sure that there are different zones that all get represented. And then finally is systematic sampling, where you will be aware that there are changes taking place across a habitat. So a good example of that happening, a habitat here is sand dunes. You can see from the sea, these sand dunes have formed because of the onshore breeze that comes off the sea. And as small bits of vegetation have formed, these little pioneer species, they've trapped sand because there's like a little wind block, and the sand has built up. And as the sand builds up, there becomes more of a shade to the wind, a protection from the wind, which makes it easier for other plants to grow, and the roots start to dig in. And as roots form, you're starting to get a form of soil forming. And look, as you go further back, it's getting easier and easier for more plants to be successful, and you've got this forest suddenly occurring at the back. So because you see this change happening, you could set up a line transect that starts from here maybe, and you run it all the way up. It might be 100 meters, and you might want to see what's happening every 5 meters. What different species am I seeing? How is it changing as I get further away from the sea? So some of the equipment that you need to just be aware of are, quadrats. Is that some of that little squares, like 50 by 50 centimeters or a meter by a meter, and you can count each square. Sweep nets, which you just sweep across. You might want to try and find some different organisms in a tree, or you could use sweep nets in a pond where you do like a figure of 8 sweep motion, which we'll go into more detail anyway. Pooters are like a little cup with a tube coming out of it, 2 tubes, and one is used to suck, and the other one brings like a little insect, and you can trap it. Point frame, it's similar to a quadrat, but rather than just chuck it down the floor, it looks like this. It's got these needles, and you place it down in, either you, I guess, are putting it where you want to put it, or through random sampling, it's been placed, and you see whatever makes contact with those pins, you count as something that you found. And there's usually 10 needles, so each needle represents 10%. The final one for measuring, so that's a big one, isn't it? There's a lot. There'll be a lot of information we still need to go through with regards to species biodiversity. This is the biggest part of this unit. The next part, though, is genetic biodiversity, and this is about looking at one individual species at a time. What is the genetic diversity of this particular species? So how genetically diverse is the species of dogs? There's only one species of dog. There's lots of breeds. Why are there lots of breeds of dog? Because there are many alleles within that species. So lots of alleles bring about lots of variation, lots of different characteristics. And that's going to prove really beneficial to any species with a high allele frequency will be much better equipped to adapt to any change. And this here is talking about natural selection. Organisms, a species, not individuals, a species, a whole population with many different alleles is much more resilient to changes in the environment. So an allele is a version of a gene, and so you might remember things like you could be homozygous recessive or heterozygous. Some genes have 3 or more alleles, and we call those locations polymorphic. So we're interested. This tells us there's potentially a lot of versions of that one gene. There's lots of different characteristics coming out of that one gene. That's healthy for that species. So we can figure this out as a calculation, another equation, to work out the proportion of polymorphic gene loci. How many gene locations on our chromosomes show 3 or more versions. So we have to just do number of polymorphic gene loci, or you could say number of genes which have 3 or more alleles divided by the total number of loci, or the total number of genes. So just think for a species which its genome might be something like 10,000 genes. It's got 10,000 different genes, or you could say 10,000 loci. And within those 10,000 genes, 500 of them have 3 or more alleles for those genes, or 500 of them are polymorphic. So what percentage of this organism has polymorphic gene loci? We'll do 500 over 10,000 times by 100, and that'll give us 5%. So again, just an overview. Go back to this when we're looking at biodiversity, be seeing the bigger picture. When we look at biodiversity, we're interested in how many habitats are there within a particular area, how many different species are there, and how even are the numbers, and how many different, what's the allele frequency of each individual species.